All right, well, I'll get us started for the group of us that are here. I've been given the privilege of introducing our speaker today. My name is Chris Kroenke. I'm at the Oregon National Primate Research Center, and I understand that this seminar series was underway during pandemic times, and it's taken a pause, but now is starting back up. And our first speaker in this series is Dr. Paul Buehler from the University of Maryland. Let's see if I can advance my screen. So this series is a combination of academics as well as industry, covering various topics in EPR imaging and oxygen detection in tissue. And today I'm looking forward to hearing Professor Buehler present to us applications of tissue oxygenation measurements in mouse models of anemia to inform transfusion quality. Dr. Buehler is a pharmacologist and toxicologist who utilizes and applied mechanistic approaches to understand aberrant red blood cells biology. And in 2019, he moved his laboratory from the FDA to the Center for Blood, Oxygen, Transport, and Hemostasis to study markers of oxygen homeostasis and erythropoiesis with oxygen measurement techniques. And thank you for participating. And Paul, I'll turn this over to you.
Thanks for the introduction, Chris. I appreciate it. And Mrignani, thank you for the invite. So what I wanted to talk about today is some of the approaches that we're taking to look at tissue oxygenation, primarily based on transfusion. I've been trying to set up various techniques here at the Center for Blood Oxygen Transport since I moved from FDA, and I've worked with several people in the field to do that. So basically, I'm going to give some background on red blood cell physiology, how red cells are stored, how they're transfused, why they're transfused, and some of that is a little bit difficult to understand and really doesn't make a whole lot of sense, but we'll kind of go through this. So, so red blood cells in general make up 83% of the cellular mass of the body, really 35 trillion RBCs per individual body in healthy people. Human RBCs have a lifespan of about 100 to 120 days. Really, we spend a huge amount of energy on a daily basis just generating 2 to 3 million new RBCs per second, 200 billion RBCs per day. Essentially, RBCs don't have any transcriptional function. But in essence, the ability of these cells really to interact with the entire system is incredible, and they do have a significant effect on the phenotype of living system. If you look at the overall production of a erythrocyte, it goes from the pro-erythroblastic stage all the way through many cellular types in the bone marrow when it's finally ejected from the marrow itself as a reticulocyte, and then matures into erythrocytes. When there's anemia, usually there's more reticulocytes that are ejected from bone marrow, and they can mature relatively quickly. And as you see here, this is overall the amount of hemoglobin generated and the amount of mRNA lost, or RNA lost from red blood cells as they're produced over time. And of course, the main function of red cells as far as transfusions is to carry and deliver oxygen. And there's approximately 250 million copies of hemoglobin within a single RBC. So when I think of overall the red cell itself, I take into account not just erythrocytes, but all the cells which are part of the bone marrow, which we just discussed. And this is basically an organ system called the erythron, and it mainly functions as gas transport, but also has function in ion homeostasis, vasodilation as far as interactions with nitric oxide, ATP, as well as the vasculature. Overall system metabolism, glucose, redox, glutathione, amino acid uptake. Also, it has detoxification function, immunomodulation function, and one of the more interesting things with red blood cells, I think, that people don't really consider is crosstalk with other cells and how they impart a phenotype on the living system and how the living system imparts a phenotype on them. You can tell a lot about a blood cell's health from particular disease states, and they are phenotypically cross-talking between themselves and other cells. When you take red blood cells out of circulation and you store them as you would in a blood bank and prepare them and ready them for transfusion, this is done in a way that is essentially regulated by FDA, and the overall practice for doing this, of course, is red cells are life-saving therapeutics. Blood banks keep them and use a lot of them within hospitals, and now they use them even in transport to hospitals as whole blood in some circumstances. But red cells can be stored under refrigeration, according to FDA, for 42 days. And this also applies in some senses to heavily processed RBCs, like washed RBCs, and potentially someday pathogen-reduced RBCs. But there are a number of changes that go on within the RBC that are, functionally change them, and overall this can affect both perfusion and oxygenation. So the things that I'm interested in, particularly as far as metabolism, are decreases in ATP, 2,3-DPG, which are critical for oxygen allosteric regulation and hemoglobin, changes in redox biology, increases in superoxide GSH and lipid metabolism, as well as alterations to the cytoskeleton itself, things like band 3, spectrin, ankyrin, as well as surface cell molecules that are expressed as a result of storage, such as phosphatidylserine and a particular interest in glycophorin A. So each of these kind of categories has an effect on both oxygen affinity, microvascular blood flow, and removal of the cells from circulation, either by the spleen or in some cases by the liver. So during storage, as I said, the issue of there's a reduction in ATP, a reduction in 2,3-DPG, which are quite severe, particularly in the case of 2,3-DPG. The pathways that generate 2,3-DPG are really inhibited by pH, low pH, as well as low temperature. And it takes time for these things to return when they're in circulation after they're transfused. So it takes hours really for ATP to return. It takes multiple hours for 2,3-DPG to return, and sometimes even days. But if you need oxygen quickly, it's going to take some time for that to happen. So always curious about this and interested in this in massive transfusion settings when people are being transfused many units of red cells and need oxygen quickly. And of course, each of these components has a different effect on the T state or R state of hemoglobin. Here's ATP and 2,3-DPG, particularly shift the oxygen equilibrium curve to the left, while increasing these two molecules drive a T state or a oxygen offloading. What else is critical here is that red cells undergo morphological change. This is a critical perfusion issue. Here you see RBCs flowing through a arteriole. These RBCs are coming close to what's called the cell-free layer, which is about 2 millimeters or 2 micron in thickness, but they never really cross into it. What's interesting, really, is that all of these cells after storage or even in circulation have different conformations, and they all go through different types of passage through the blood or through the capillary, through tumbling, trembling, tank treading, stretching. Stretching is a normal response. Tank treading, trembling, and tumbling all have somewhat pathological responses if they occur excessively. And if you look at red cells in general under storage and how they affect flow, the primary effect is really that red cells increase margination toward the vasculature and then at some point start to impede flow. And this can be looked at really in the sense of a functional capillary density, where you're looking at the number of perfused capillaries per unit area. Let me see if I can get this to work. No, guess not. Okay, so essentially the cells have to go through a very, very small area, and they have to deform to be able to do that. If they can't do it, you basically lose the function of the capillary. And as you lose more of these, the perfusion obviously to tissue decreases. And as you can see here, these are red cells going through a capillary deforming. If we were to stop this, if you were to plug up this capillary, you would lose the functional capillary density. But they're quite efficient at doing this under normal conditions. So what is really the criteria of quality of a red cell during storage? There's two metrics that are put forth by regulatory agencies. One is called post-transfusion recovery. This is essentially a issue where you inject a radiolabeled red cell, and then you follow it over a 24-hour period. What regulatory agencies like FDA say is you can't lose more than 75% of those red blood cells over a 24-hour period. So, if you get something like this down here, this is an unacceptable PTR. You would not be able to transfuse such a cell. And acceptable PTRs are up over 75%. And there's some PTRs that fall below this, but are still considered acceptable. So this is where we get into kind of a gray area with the quality of a red cell. The other issue is the percentage of hemolysis within a storage unit. So we allow 1% of hemolysis within a storage unit. You can see this is greater than 1% based on a calculation of hemolysis. And then this would be acceptable based on less than 1% hemolysis in the blood bag. And this is really kind of all we have to judge how red cells really function during storage or what their quality is. So hemolysis itself in vivo or in circulation in a living system is essential to biology, really, because we are always clearing red cells. This typically happens through the spleen macrophages, red pulp macrophages. But when we do this in terms of looking at post-transfusion recovery, it's largely affected by what the morphology of the red cell is. And this is a basic viscosity shape of a red cell. As it ages, you see increasing changes within the red cell membrane. This is starting to become a pre-echinocytic cell, a echinocytic cell, a late echinocytic cell, and what is finally called a spherocyte, which is a very rigid red blood cell with very little deformability. If this were to go through a capillary, it would basically plug the capillary and you would basically lose the function of that capillary. So as I said, the way you do this is basically by radiolabeling the red cells. And then the way we think about this is we have some donors that are good stores and some donors that are poor stores. Around this 75% allowable post-transfusion recovery, we have people who have their red cells circulate for a longer period of time and they can be recovered over a longer period of time. And then we have other donors who have generally poor storing red cells, and they're termed poor stores. So what is the reason for this? Really, this has to do with the donor themselves. Really, it's an interesting process by which, in many times, donor genetics have a large impact on the quality of red cells during storage. And this is something that's really been looked at by NIH over the last 10 years, looking at the recipient and donor project, where classify donors as being either good, poor stores based on what their genetics are and how that affects red cell function. Many of these GWAS identified polymorphisms within individuals lead to issues as far as oxidative stress within red cells that cause them to store poorly, and then as a result transfuse poorly. And there's been a lot of work on this, and much of this, there's a large body of information in the literature on donor genetics and how this affects red cells. What else is incredibly interesting, I think, is that not only are red cells affected by genetics of the donor, they can also be affected by what the donor is exposed to. For example, things like diet, intake of nicotine, caffeine, ethyl alcohol, prescription drugs, exposure to pollutants, what the diet is, if the person exercises, what's their microbiome. Age has a very significant effect on this, as well as stress and BMI. So there's been a few very interesting studies by one group at the University of Colorado Anschutz that has looked at both the genetics and how this affects donor storage quality, and has broken this down into many factors that affect the actual storage of a red cell and the transfusibility of a red cell based on factors that affect the exposome. And we can then begin to start taking some of the things we see with, for example, the genetics and the exposure factors in donors, and start modeling those in other systems such as mice. And one way to do this, obviously, is to look at the post-transfusion recovery, which I think is a bit rudimentary, and we'll talk about going into methods to measure oxygen. But for example, here, B6 mice actually store very well. You can recover almost all of their red cells after a transfusion. Whereas if you took, for example, an FEBJ mouse, these mice store very poorly. There's actually an interesting reason for this. FEB mice are high expressors of STEAP3, especially within their red cells, which is a ferroreductase, which actually leads to increased reactive oxygen species within the red cell itself. And we've looked at this with Joe Gao here at the University of Maryland. And if you consider, very basic functions within the red cell, particularly those within glycolysis in the pentose phosphate pathway, whereas glycolysis, you have production of NADH, whereas the pentose phosphate pathway, you have production of NADPH. Critical to this is that you always have an ongoing autooxidation of hemoglobin, which is this cycle of iron 2+ state to 3+ state hemoglobin, one of which binds oxygen, iron 2+, one of which does not, and one which becomes quite unstable, which is hemoglobin 3+. And there's methods, obviously, within the red cell to handle this through cytochrome B5 reductase or methemoglobin reductase, and then NADH is a cofactor. Obviously, this process leads to superoxide as 2+ or oxyhemoglobin oxidizes to methemoglobin and then forms superoxide, and this can be measured essentially using, in this case, CMH. CMH then can be detected after reaction with superoxide to identify the nitroxide here, which then essentially, we can identify the production of superoxide within the red cell, which is actually, it's important to identify this, I think, because as the overall reductive capacity and the oxidative insult to a red cell progresses during storage or even disease, you start to form even more complex species of hemoglobin, where you get not only oxidation of the iron itself to +3, but you start to form aberrations within the porphyrin itself, which can then form things like hemochrome, Heinz bodies, which bind to the membrane of the red cell and lead to issues that directly cause hemolysis, either intravascularly or extravascularly. And if we take a look at the FEB mouse versus the B6 mouse, which have genetic differences, the FEB is a bit predisposed really to the formation of oxygen radicals such as superoxide. FEB mice demonstrate CMH oxidation at a much faster rate and indicate a higher steady state level of superoxide once you take them out of the body and store them over multiple days than FEB mice, which do not have direct aberrations in their genetics that would predispose to a oxidative stress within red cells. So overall, you can start to model these things in animal species, particularly mice, that could mimic much of the genetic changes that donors have, which are imparted onto the red cells, which can be used during storage and transfusion. So the idea then is to have a precision medicine type approach to understanding these things. Again, how do we really transfuse red blood cells, and how do we make such decisions? And this is an example of a recent trial, which called the MIN trial, which is myocardial ischemia and transfusion, which was performed to look at what would be called a liberal transfusion range, which is greater than 8 but less than 10 gram per deciliter of hemoglobin, or a restrictive transfusion strategy where there's a hemoglobin of between 7 and 8. And the question's always been really, when do you start to transfuse patients? the issue of doing that has been more of a clinical judgment. And if you say at 8 or less than 10, greater than 8 that you transfuse, your hope is that 1 unit increases the RBC, the hemoglobin mass of that 1 unit of RBCs by 1 gram per deciliter. That's what is expected. So what people then start to look at is how much is the incremental change in hemoglobin after a transfusion of the number of units of red cells that they give. So to get this person back up to a normal hemoglobin of, say, 12 to 15, you'd need multiple units, and hopefully you could get that up by 1 gram per deciliter. However, typically the incremental change is not the same as the expected, which would require then additional transfusions. What I think is absolutely amazing is that we never consider the issue of measuring oxygen in tissue, which is the main function of red cells in a transfusion setting. Their primary function, obviously, is to deliver oxygen, and that's what they're indicated for during transfusion. But we do not measure that, which is amazing, because if you're treated with an antihypertensive, you get your blood pressure checked and dose adjusted based on that. Where here you're basically using a clinical judgment kind of blindly to make assumptions about whether to transfuse again or not transfuse again. And I made this argument when I was at FD. I said, why don't we measure oxygen instead of things like post-transfusion recovery and hemoglobin incremental changes? And at that time, there's a clear reluctance to approach these things when you don't have an idea how to deal with them, but you really have to start somewhere. So where I started was meeting Hal Schwartz and being able to go up to Dartmouth and do some experiments to kind of prove to my office director in the Office of Blood that these things might be a good idea, to be able to at least start looking at them in a preclinical setting, and over time maybe these things could be applied clinically. So what we did was a very simple study. We started looking at oxygenation to muscle tissue. This is not a mouse, this is a rat, but it's still under the murine species. So here we started looking at chip implantation with oxychip, and we didn't use the M-chip, we used the oxychip here, and that was implanted into the hind limb muscle, the gastrocnemius of the rat, and then we basically looked at oxygen changes based on hemorrhage. So in a initial phase of this experiment, the timeline is this. We looked at baseline oxygenation, then a period of blood loss with an oxychip implanted in the gastrocnemius here. Withdrew blood to decrease mean arterial pressure. So this is really a pressure-controlled hemorrhage to about 30 millimeters of mercury, and held that over a fairly short period of time, 30 minutes. And then we could measure through oxychip the drop in PO2 within the hind limb, and get a fairly decent response, really, with a nadir PO2 of about 5. In these animals, the blood loss was very consistent across groups. We looked at animals that were going to be grouped into 1-day, 7-day, and 14-day stored RBCs, and then a albumin group to control for no oxygen. And just to I don't want to make this assumption, but most species such as rats and mice, it's been worked out over the last several years that approximately 14 days is the maximum that you can store a mouse or a rat red cell to approximate or translate to the human equivalent of 42 days storage. So that's the basis for looking at this range. Then looking at the transfusion and the post-transfusion, which we transfused red cells back at those different storage time points. And what we observed here is that the blood pressure returned in all circumstances at baseline, and then the drop, and then the increase post-transfusion. Similarly with hemoglobin, saw this decrease, which is expected. Then an increase in hemoglobin after transfusion of the 1 day stored, 14 days stored. Of course, we saw this with the 7 day stored as well. Albumin did not increase as expected. But what is interesting, though, is that even though we get a restoration in the overall hemoglobin, what we don't see is a restoration in muscle PO2, which is an interesting thing because we base everything on hemoglobin concentration or hemoglobin concentration within circulation to transfuse people. And that may not be the best idea if the hemoglobin goes back to what you would expect or goes back to a level that we believe would be beneficial and you're not oxygenating certain tissues. This can be seen here at 7 days, and it's even seen almost similarly at 14 days. So that would suggest that you're starting to lose and equivalently losing function at 7 days compared to 14 days. And this, quite frankly, isn't that much better than albumin. So after that study, we started looking at different methods after I moved to Maryland, and I'll give an example of a couple of these. And this is where we've really started looking more at the mice in terms of being able to start to look at genetic differences, which we're doing now. We have several studies ongoing, but I'm just going to explain what we were doing with basically with B6 mice. And we have a blood bank for mouse blood itself. We use donor pools to take blood from, process that blood as you would in a blood bank, store that blood as you would in a blood bank for various days. And for example, we can then characterize that blood in terms of its functional capabilities before we transfuse it. And for example, here's an oxygen equilibrium curve in its bulk form. So at day 1, the oxygen equilibrium curve is quite normal. The P50 in a B6 mouse is a little bit high. It's about 46 to 49, which is expected. But as storage goes on to day 7, day 14, this starts to left shift as you decrease 2,3-DPG, to some extent ATP, and the overall P50, the overall oxygen saturation decreases. So this is a bulk measurement of red cells. What we're also looking at with a collaborator at Harvard, John Higgins, is how to break down within these bulk samples of red cells what the oxygen saturation is per mass of hemoglobin, to understand within the stored red cells what is potentially has a optimal oxygen saturation and what doesn't, and what can bind oxygen appropriately and what can't. And then there's some interest in being able to modulate these red cells in terms of adding things back to be able to improve the allosteric response to hemoglobin. But it is very curious that you get populations within these bulk samples where you have different oxygen saturations. And then we look at morphology of these red cells, and as you can see, they change quite dramatically over time. Here's day 1, day 2, and day 3, and by day 3, you're already out to very, very rigid cells, which are consistent with what are called the spherocytes, which are the very round, rigid cells that don't passage capillaries very well at all. One technique that we've looked at just to, be inclusive in this process overall of understanding tissue oxygenation is phosphorescence quenching. We work with, to some extent, Sergei Vinogradov at Penn, and we've used his oxy-4 compound to look at oxygenation to liver. And what we did here, and this study is really hypothesizing that hemoglobin increments do not actually predict what the oxygenation is to a tissue. So if you see a hemoglobin that's basically normal after you do exchange transfusions, where we did, in this study, the way it's set up is we did multiple exchange transfusions over multiple time points to essentially try to replace approximately 80% of the overall blood volume with transfused blood. And that transfused blood was at either 1 or 7 days of storage. And that blood was transfused back into these animals at a 45% hematocrit. So ultimately, the stored blood was returned to the animal to replace approximately 80% of their circulating blood volume. Okay, so the interest then was, what's the change in hemoglobin over time with the different storage times of red cells? And here's day 1 stored and day 7 stored. The hemoglobin is actually quite close. Albumin, obviously, this drops pretty dramatically. And if you look at the total hemoglobin at day 1, day 7, they're different, but they're not dramatically different. You look at the AUC, these are also quite similar. But what you do notice, obviously, is then a very dramatic drop in the PO2 of the tissue, such that this is not predicting this. So overall, the issue of looking at things related to total hemoglobin are not predicting what you're seeing in the tissue itself. So again, we've moved on to looking at imaging, from oximetry to PE quenching to EPR imaging, which I think is useful for us in many ways. And we worked fairly extensively, very extensively with O2M and Murignani and Boris to really implement this technology here. And I think it's nice because then we have multiple ways to look at tissue oxygenation as it relates to transfusion, particularly in our disease models. But we finally set up a system within the last year and a half with the instrument that we purchased from O2M. And basically the model that we've set up is doing a fairly extensive catheterization in mice, and then being able to transfuse these mice by then first doing basal imaging by EPR, then hemorrhaging animals for approximately 15 minutes. This is a fairly extensive hemorrhage, so it's approximately 60% of the blood volume. And these animals are basically, at very low mean arterial pressures. They have, can maintain them for a very short period of time under these conditions. So it would be like really a massive blood loss, which would require a large volume transfusion. So then we can image after the hemorrhage itself. We can then resuscitate with different types of types of red cells, in this case, stored red cells, 1, 7, and 14 days. I put this in here because we often look at artificial oxygen carriers. And then obviously we're using OXO 71 as our paramagnetic agent. So some of the data we've been able to gather, this is just a simple hemorrhage and retransfusion of different storage times at 1 day, 7 day, and 14 day. Here we see very substantial decrease in oxygenation to the kidneys. And then depending on the age of the red cells, we either see a percentage restoration of the oxygenation, which is almost linearly time dependent on the storage. And we can then start applying these to our other models. But from this standpoint, I think looking at the types of things we looked at with this type of imaging, we can extend it to other tissues as well. And then obviously having it coupled with anatomical structure and MRI, it's useful from the standpoint of doing kind of multi-tissue analysis. And then looking at a three-dimensional approach to the oxygenation throughout tissue. For example, here at baseline, at 1 day of storage, 7 day of storage, 14 day of storage, there's fairly dramatic differences within the overall oxygenation to the kidneys. One thing I will say is, because I think right now we're in a preclinical stage of trying to really develop things that I think would be useful to translate to clinic, because ultimately that's where we need to be. Unfortunately, some of the things that we're looking at right now may or may not tell us, what we need to know. But even from a research-based standpoint, this is kind of what we have now. Basically understanding the kidney as a sensor of hematocrit or hemoglobin levels and overall hypoxia, or its detection of hypoxia as related to stabilization of hypoxia-inducible factors. I'm particularly interested in HIF2 because that's the primary transcription factor for EPO and the onset of erythropoiesis, which you should not have with a quality transfusion. And obviously, EPO has its effect on EPOR receptors within the bone marrow and production of red cells. And we can look at various other hormones such as erythropoietin, where EPO, when it's increased, erythropoietin is increased and hepcidin is decreased. So there are types of biomarkers which we can use now, but where we obviously want to be is directly measuring oxygen in these tissues, particularly the kidney. And then here's just some data that kind of supports the idea of looking at selected biomarkers currently in a research setting to understand if these could be used in the setting of clinical transfusion. And I think that this is fairly simple to do, but as I said, where we want to be is to actually measure oxygen in these tissues. So what are my thoughts and conclusions? And this is really that in a preclinical setting, we can learn a whole lot from mice based on potential for inducing genetic diversity just on its own, or doing this through transgenic mechanisms. And we can basically model anything that we see in the human in terms of genetic background. We can expose animals to basically anything related to the exposome and look at red cell function in terms of how we transfuse a recipient. Nonetheless, there's really important limitations to both the technology by which we measure oxygen and the animal models that we're using. So I think within the preclinical setting, it's working well right now, but where we really have to take this in a preclinical setting, and I talked to a lot of people who are working in research-based areas in transfusion, and what they want is something that can be used very easily with not a lot of back-end analysis necessary, with, say, co-registration of oxygen and anatomy. And it needs to be universal across labs, because this is something that there's some concern with from the people that I talk to and work with. And in the far future, it would be fantastic to have this in a clinical setting, really to understand if RBC transfusion is needed in a certain patient, and what's the effectiveness of that transfusion in a patient. And really, to understand this, we can only do it by measuring oxygen. Otherwise, it's really a guess. And with that, I'd like to thank the people in my laboratory. I'd like to thank Joe Gao, Eric Legenzov, who I work with very closely, especially on imaging as well as oximetry and trying to measure free radicals as well as reductants in tissue. And then obviously all of the people at O2M, ClinEPR, Sergey, particularly the people at U of C, Howard, Boris, and Daniela, who helped us out quite a lot with potential to use implantable resonators and issues surrounding that, so we can do multiple tissue oximetry. So I want to thank you for your time, and I want to thank you for listening to this. And I hope there's things that we can do in the future to advance a complex field like transfusion medicine, which I think needs a lot of help on the side of being able to really understand the products at which they give to people in terms of effectiveness. Thanks.